Hello, everyone. My name is Jermaine Mystery, and I'm a chemistry major here at TCNJ. I'm a student in Dr. Benogget's lab, and today I'll be sharing my work testing Ctab reverse micelles to study late embryogenesis abundant proteins. So I'll first be giving a general overview and then going a little bit more in depth. So my general overview. We are studying group 3 LEA or late embryogenesis proteins found in soybeans. And these proteins are part of a large, broader group of proteins called intrinsically disordered proteins. And these proteins do not have a specific three-dimensional structure under aqueous conditions, but when put under stress, such as dehydration or extreme pHs or extreme temperatures, they do form a defined structure. So the same applies to our group 3 LEA peptide or G3-LEA. And in previous studies using circular dichroism, they found that these peptides had a helical structure at low temperature, and as temperatures increase, they were losing their helicity, which was expected as proteins tend to denature as temperature increases. But at about 55 degrees Celsius, they gained helicity. So that means they were forming structures again. And we suspect that these proteins were dimerizing to form coiled coils. Then after that, Forster resonance energy transfer, or FRET, was used to further study this dimerization. But there was a problem. When the data came through for the FRET, there was a large unexpected band in the graph, so as you can see here. And so we found out that the reverse micelles that were used in the solutions for FRET, and these reverse micelles were used to stimulate conditions of low peptide hydration, they were interfering with the FRET. And so the solution we came up with was to make reverse micelles out of another substance, and that substance we chose was cetyl trimethyl ammonium bromide, or Ctab. And so the goal of my work was to see if Ctab would be a suitable substance to use for reverse micelles for our purposes of studying these G3-LEA proteins. And our results so far have indicated that yes, it does work. To go a little bit more in depth about FRET and how it works, basically FRET is a method where energy is transferred from one molecule to another, and the amount of energy transferred is dependent on the separation between those molecules. And so we tagged these G3-LEA proteins with cyanophenylalanine, or FCN, and tryptophan, or TRIP. And basically, FCN is excited at 240 nanometers and emits at 280 nanometers, and TRIP is excited at 280 nanometers and emits at 350 nanometers. So their emission and absorption spectrums overlap, which makes them a good pair to use for FRET. And so when AOT was being used as a reverse micelles, it was absorbing some of the energy that was used to excite FCN, and it was also absorbing some of the energy that was being emitted from FCN. So it was not fully transferring its energy to TRIP. And so that would affect our FRET, our calculated distances using the FRET information. So that leads to incorrect results. So for my reverse micelles, the Ctab reverse micelles, we performed scans to ensure that it did not absorb and emit at the same wavelengths that FCN and TRIP do, so that it would not interfere. So to do that, we use UV spectroscopy and to determine the absorption spectrum, and we use luminescence spectroscopy to determine the fluorescence emission and excitation of the Ctab reverse micelles. Like I said before, our our Ctab reverse micelle scans came back, and it looks like around 290 nanometers. Ctab shows no absorbance, and that is where it would interfere with VCN energy transfer. And it shows very little to no emission excitation at wavelengths where VCN and tryptophan emit light and are excited. So that indicates that CTAB could be used. And so for our future work, we would have to also look at run some FRET scans of TRIP in the CTAB reverse micelles, as we've only used VCN so far and did not have the chance to work on TRIP yet. But if that all goes well, then another thing to consider is pH, as pH is an important factor in protein structure and function. So in the future, we're looking to be able to control the amount, or control the pH in the reverse micelles, so that we can control it and make it simulate cellular conditions. So that's it for my presentation. I would like to thank Dr. Benoggin for her guidance and throughout my research, and the members of this lab who have performed all of these previous experiments to lead to my project today. And I would also like to thank the TCNJ Chemistry Department for the equipment and space for facilitating this project. Thank you for watching.